Welcome back to Dr. Liu's VCE Biology. Today's topic is the disease challenges and strategies, as presented in the 2024 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The pathogen causing beak and feather disease contains single-stranded circular DNA, a feature characteristic of viruses. Therefore, this pathogen can be classified as a virus. During an allergic reaction, activated mast cells release a variety of chemical mediators, including histamine. Histamine is the primary substance responsible for many of the classic allergy symptoms, such as inflammation, itching, and swelling. Not all bacteria have a cell wall, but those that do can be classified as either gram-positive or gram-negative after undergoing the gram staining technique. Pertussis is a highly infectious disease, caused by the gram-negative bacteria Bordetella pertussis, which is transmitted through respiratory droplets when people cough and sneeze. Individuals with suspected pertussis infection can have their throats swabbed. The swab will contain all the types of bacteria present from that region of their throat. Gram staining can then be performed on these bacteria. Gram staining is a technique used to identify bacteria based on their cell wall. Bacteria with a thick cell wall are gram-positive, staining purple. Gram-negative bacteria, like Bordetella pertussis, have a thinner cell wall and do not retain the purple stain. They appear colorless or are counterstained pink or red with safranin. It is important to note that gram staining is not a DNA amplification method. The use of ethyl alcohol is a key step in the process, not something to be avoided, as it differentiates between the two types of bacteria by washing away the purple stain from gram-negative cells. The entry of B. pertussis into the body initiates a robust immune response. Subsequently, dendritic cells become activated and migrate to the lymph nodes to present B. pertussis antigens, a crucial step for the adaptive immune system. The spread of B. pertussis can be most effectively limited by establishing herd immunity with the available vaccine. Multiple sclerosis, MS, is a disease in which the immune system damages nerve cells in the brain and spinal cord. Alemtuzumab is a monoclonal antibody that has been shown to be effective in delaying the progression of the disease in some people. It is thought that alemtuzumab binds to a protein known as CD52, which is present on the membranes of B cells. This leads to B cell destruction by activating complement proteins. The complement system can eliminate B cells through three primary pathways. Membrane attack complexes, MACs, formed by complement proteins, can induce lysis of the B cell plasma membrane. Opsonization occurs when complement proteins coat the B cells, leading to their subsequent phagocytosis. Complement proteins also function to recruit inflammatory cells that aid in the destruction of B cells. Multiple sclerosis, MS, is a disease where the immune system attacks nerve cells in the brain. Since B cells are key to this process by secreting antibodies, their destruction leads to fewer autoantibodies, less nerve cell damage, and a reduction in MS symptoms. Because T and B cells share the antigen CD52, and because the variable regions of alemtuzumab are complementary to this antigen, the antibody can effectively bind to either cell type. Rabies is a serious viral disease that is usually spread through the infected saliva of dogs, bats, and foxes, but all mammals are capable of contracting and spreading the virus. Treatments for rabies in humans include: Treatment 1. a vaccine that contains the inactivated rabies virus. Treatment 2, injection of specific antibodies directed against human rabies. Administering these two treatments at the same time is effective because they provide different benefits. The first treatment is advantageous for its ability to stimulate the production of memory cells, which ensures long-term immunity. In contrast, the second treatment provides immediate protection by supplying antibodies that can directly bind to the pathogen, helping to reduce the severity of rabies symptoms. Individuals who frequently travel to areas where rabies is prevalent are advised to receive a vaccine followed by a booster after 3 years. The rationale for this is that memory cells and antibodies diminish in number over time. The booster dose serves to stimulate the immune system, leading to the production of additional antibodies and memory cells. Consequently, this allows the individual to mount a quicker and more robust immune response upon subsequent exposure, thereby reducing the likelihood of developing rabies symptoms. For almost a century, Europe has been free of diseases carried by mosquitoes. Recently, mosquito species that can cause disease are becoming increasingly common in Europe. Of particular concern is the Asian tiger mosquito, Aedes albopictus, which can transmit deadly viruses such as dengue, chikungunya, and Zika. With global warming leading to warmer temperatures and higher rainfall in Europe, it is predicted that these mosquitoes will continue to move further north every year. The transmission of the chikungunya virus via mosquitoes can be controlled through a multifaceted approach. This includes the isolation of infected individuals, public education on control and avoidance measures, the removal of mosquito breeding grounds, and the use of vaccination to achieve herd immunity within the community. An endemic disease is characterized by its persistent presence within a specific population, with the number of affected individuals remaining stable and under control. An epidemic disease, however, is marked by a sudden, rapid increase in the number of cases within a short time frame. Until recently, a vaccine for the Zika virus was not available. However, a treatment using monoclonal antibodies, mAbs, has proven effective in reducing the viral load in pregnant women and mitigating the effects on their unborn children. The production of these antibodies involves a process where an animal is immunized with a viral antigen to elicit an immune response. The resulting antigen-specific plasma cells are then harvested and fused with an immortal tumor cell line to form a hybridoma. This hybridoma cell can then be cultured to synthesize large quantities of the desired monoclonal antibodies, which are subsequently isolated for clinical application. Both antigenic drift and antigenic shift cause changes in viral antigens, but they differ significantly in scale and effect. Antigenic drift is a minor change resulting from a point mutation in a single protein, which creates a closely related strain that typically affects the same species. Antigenic shift, by contrast, is a major genetic change caused by viruses swapping whole sections of their genomes. This can produce an entirely new virus strain and potentially allow it to infect a new species. An individual's prior exposure to influenza, whether through vaccination or infection, enables the production of antibodies and memory cells that target the virus's surface antigens. However, if the antigen's shape undergoes a change, the person's existing antibodies may no longer be complementary and will be unable to effectively bind to the new viral antigen. Consequently, this leads to a loss of immunity to the new strain. This exam section, worth 32 marks, 26%, covers 5 main topics related to diseases and the immune system, multiple choice questions, 5 marks, questions 24 to 26, questions on how viruses are classified by their DNA, AQ24, how bacteria are identified using gram staining, Q26, and allergic reactions, Q25, questions 27 to 28. Questions on the roles of adaptive immunity and herd immunity. Short answer questions, 27 marks. Question 5. A short answer question on multiple sclerosis, MS, and how the drug alemtuzumab targets B cells to reduce symptoms. Question 6. A question on rabies, covering the use of vaccines and antibody treatments, and the importance of booster shots to maintain immunity. Questions 7 to 8. Questions about controlling mosquito-borne illnesses like Zika and Chikungunya, Q7, and a question differentiating between the two types of viral evolution, antigenic drift and antigenic shift, Q8. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.